Hello, class. So this is our first topic for prelims, the microbial world and you. For our topic outline, we have our overview, microbes in our lives, naming and classifying microorganisms, a brief history of microbiology, microbes and human welfare, and as well as human disease. For our learning outcomes at the end of this topic is you will be able to differentiate the major characteristics of each group of microorganisms. Second, understand impact of microorganisms on human affairs. Third, explain the development of microbiology as a science and different contributions made. And lastly, recognize the beneficial activities of microorganisms. So microorganisms is also called as microbes, which are organisms that requires microscope. So we all know that microorganisms are very minute organisms that we can only appreciate their structure with the help of microscope. So without microscope, we will not be able to see their morphological structure, no, in order for us to identify those structures with this kind of microorganisms. So with that, microbes are also said to be dominant organisms on Earth. They're considered to be the oldest type of microorganisms on Earth, especially that if ever there are microorganisms trapped in ice, like for example in Antarctica, that is really possible that there are microorganisms that are trapped with that type of habitat or environment. That is why it is said that microorganisms are said to be dominant organisms on Earth and the oldest one, because there are also records of microorganisms that plays important role as that helps in the cure of certain diseases during ancient time. That is why microorganisms are said to be dominant and oldest organisms on Earth. Aside from that is these microorganisms have major groups, no? And these are bacteria, algae, protozoa, fungi, parasitic worms or the parasites, and viruses. So these are major group of microorganisms considered to be distinct with each other. They also have own characteristics and own habitat, no? So we will have a glimpse of these different major groups of microorganisms. Aside from that, we need also to consider different fields of sciences in order for us to know and understand the science of microbiology. And these are genetics, immunology, biochemistry, epidemiology, and ecology. So these different fields of sciences are very important in order for us to know more the world of microbiology, no? Especially in terms of cell structure. Okay. Aside from that, microorganisms plays also a very crucial role, which serves as photosynthesizers, decomposers, and recyclers, because there are type of microorganisms that plays very important role in the ecosystem, no? Like for example, the role of bacteria and fungi as they help the soil in degrading inorganic to organic matter that considers to be a usable form for the soil that makes this soil more fertile. That is why bacteria and fungi are very important in the field of ecology in terms of helping degrading these nutrients, no? So aside from that, microorganisms are very important also in the field of industrial production, agriculture, medicine, and environmental protection. That is why in the study of microbiology, there are other fields of microbiology that is very important um, in our different fields of microbiology. Like for example, food and industrial microbiology, as well as bioremediation and other advanced technologies that help us to have a better way of life because also of the benefits that we can have with these microorganisms. That is why these microorganisms plays a very important role in our lives. They're not also considered to be beneficial, but as well as there are also microorganisms that are considered to be infectious or harmful to us, no? Then at the same time, in studying microorganism, we need also to use scientific approach in order to develop theories, findings, and explanation. That is why in the study of microbiology, it is really important to follow scientific approach or scientific method in order for this researches to be a reliable one. No? So that is why if you're going to conduct researches, regardless of what discipline, you need to follow scientific way of method in order for that research to be a reliable one, no? So during midterms to finals, no, I will also consider some topics, no, which is very important in the field of microbiology, which is the microscopy, culture techniques, and other methods of handling or controlling microorganisms. That is very important in understanding more about microorganisms. No, how are we going to um, how are we going to isolate? How are we going to identify this microorganism inside the laboratory? No? So we have these different discoveries and events related to microscopy, culture techniques, and how do we control and handle these microorganisms. So these are very important topics. So we will cover these topics during midterms. Then aside from that, we need also to consider the evolutionary relationships of these microorganisms, their scientific names, and their characteristics. If you can, remember when the time pandemic started, is there is only one strain of coronavirus that time, but as time goes by, microbiologists reported that aside from the first strain, there are several um, strains of coronavirus after that one. So, um, how they did do this, no? Or how did they discover this one? So, by means of advanced technologies, like for example, molecular analysis or phylogenetic analysis. These advanced technologies will help our microbiologists to determine the evolutionary relationship of these different strains of coronavirus, for example. So it looks like, imagine class, yeah, it looks like a family tree wherein you will be able to identify the lineage of your family tree. So it looks like that. So that is why it is very important to know the evolutionary relationship, because you will be able to identify if this specific strain of coronavirus is somewhat related to the next, or from the previous strain that you discovered. So that is why the time pandemic started and they discovered the first strain of coronavirus, they reported that the second coronavirus strain is somewhat related to the first one, but there is also that very distinct characteristic belongs also to the second coronavirus strain. So that is why we need to also to consider evolutionary relationship in order to understand and identify fully this type of microorganism. So therefore, microbiology is a specialized area of biology that deals with living things ordinarily too small to be seen without magnification. That is why it is really a big help for us if we know how to manipulate microscope. So in order for us to view better these very minute organisms. So that is why these microorganisms are said to be microscopic, no? So it comes from the Greek word micros, meaning small, scopen to see, and bios referring to life. So talking about major groups of microorganisms, regardless of what group of microorganisms, there are also microorganisms class that are easy or difficult to be seen under the microscope, no? So as I stated here in our slide, easy to reproduce rapidly. Perhaps this microorganism can reproduce rapidly and grow quickly, which results in large population in the laboratory. That is why it is so easy to magnify or see their population. But there are also groups of microorganisms class that are difficult also to be seen under the microscope. That is why in manipulating microscope, you need to have that patience in order to view your specimen or that particular microorganism. So that is why in the field of microbiology, it is considered to be largest and most complex of the biological sciences, because we are not dealing only the world of microbiology. You need also to consider other fields of sciences, as I mentioned earlier. And with that, you need to deal with their genetic makeup, their physiology or the function, their characteristics, interaction with their environment or habitat, interaction with their host, no? Because I mentioned earlier, we have a group in microbiology referring to parasites. Then, of course, their uses, no? The uses of this microorganism, which also very important in different industries. So how important microbes in our lives, no? So microorganisms are considered to be very important to us because we all know that in our body, we have this natural microbiota, no? Especially in our small intestines, where we have these natural microorganisms in our digestive system that plays very important role in our digestion. And with that, it helps maintain the balance of life in our environment. And we also have microorganisms that are present in different habitats, no? Like for example, marine, that can be found in marine ecosystem, freshwater, that is very important when it comes to the different functions. Like for example, food chain, no? And these microorganisms are responsible with what we call as food chain happening in oceans, lakes, and river. So the habitat of microorganisms is not only limited to one type or kind of habitat. So we can find them anywhere. That is why microorganisms are said to be ubiquitous, because they can be found anywhere, no? Aside from that, I already mentioned this one, that microorganisms are very important living organisms that helps us degrade this inorganic to organic matter to be a useful one. That is why microbes plays very important role in the fertility of the soil. Then there are also microorganisms, or there are also a type of group of microorganisms that has a function for photosynthesis. So later I will going to mention that. And then, of course, I also mentioned this one, that we have this natural microbiota in our digestive system, particularly in our small intestine, that helps in our digestive process and synthesizing these vitamins that are very important in our body. So with that, the study of microbiology, especially for food and industrial microbiology, are very important field or science in microbiology, because it enables to produce vitamins, enzymes, alcohols, drugs, condiments, cheese, yogurt, bread, alcoholic beverages. That is why food and industrial microbiologists are very in demand job, not only here in the Philippines, but as well as in abroad. So next is we will proceed to naming and classifying microorganisms. Okay, so we have this what we call as nomenclature, which is established by Carolus Linnaeus. It is a system of naming organisms. So this nomenclature is very important because it serves as a guide for researchers in order to have a systematic and organized way of identifying species or organisms. So with the help of this catalog, the researchers were able to identify these living organisms to avoid chaos, to avoid confusion. Because in the science of biology, it is very important that if you're going to have this chance to discover new species, you need to be sure that the species that you discovered is really identified well with all the characteristics, and at the same time, it is really considered as new species, no? So that is why you need to consult experts on this particular field in order to verify your claim or your discovery, no? So that is why very important itong na-discover ni Carolus Linnaeus na catalog. Aside from nomenclature, is we also have the taxonomy, which is the formal system of organizing, classifying, and naming living things. So with that, we have three primary concerns of taxonomy: the classification, which is an orderly arrangement of organisms into groups. For example, you're going to group a bacteria according to the number of flagella, no? For example, there are bacteria class that the flagella or the flagellum is more than one. You can also, for example, you can also group the bacteria according to the arrangement of, in terms of arrangement of their flagella or flagellum, no? So those are example of classification. Then we also have identification in terms of recording the different traits of an organisms, no? So aside from that is we also have the levels of classification. As you can see here, class, this is quite familiar to you, no? During high school days, for example, the scientific name, no? As you can see here in our illustration, you can see the domain until species, no? So for the domain, you have an example here belonging to eukarya or eukaryotes. So as you go down to these levels of classification, you will have a very specific taxa, no? So As you can see here in our domain, it refers to giant all-inclusive category based on unique cell type, one or few general characteristics. So as you go along, no, with eukarya, it will give you that very specific example under eukarya, no? So from domain to species, meaning general to specific, okay? So species, for example, is the smallest and most specific taxon, no? So that is why under eukarya, now, the most specific taxa that you can state here, no, is Homo sapiens, though there are a lot of examples for eukarya, okay? So that is why from domain to species, it refers to general to specific type of species. We also have here the phylogenetic tree of life or the tree domain system. So as you can see here, we have the bacteria, archaea, and eukarya. So each of this group class has different microorganism belonging to each group, no? As you can see here, the bacteria and archaea belongs to eukaryotes, no? Well, of course, the eukarya belonging to eukaryotes, no? So with that is, if we are talking about these different microorganisms class, in biology, no, we need also to consider the degree of relatedness, no? And that is what we call as phylogeny. That is why, as I said earlier, there is what we call as phylogenetic analysis, which is considered to be one of the advanced technology in microbiology and genetics that enables you to determine if this microorganism is related to one another or not. And through this advanced technology, whether molecular or phylogenetic analysis, it helps you to determine further and comprehensively if they are related or not. So just like my example earlier with regards to the different strains of coronavirus. So next is assigning specific names. So this refers to scientific names, no? So we all know that the scientific name is composed of two words, no? The first word is what we call as the genus, while the second word referring to the species, no? So the first word, which is the genus, okay? The first letter should be capitalized, okay? The first letter of the word which referring to the genus name should be capitalized. No, for example, this one. And then the second word should be lowercase, which is referring to the species name. So, for example, this one. So, if we're going to state or write a scientific name, it could be, you can also spell out the scientific names, no? Like for example, this one, or you're going to shortcut the genus name, just like this, no? Okay. But if you're going to state the scientific name, for example, soft copy, no? Like for example, on this slide, you need to italicize the scientific name, okay? Just like this. But if you're going to write it down, no? Like for example, penmanship, no? So you're going to underline, no? The two words, the genus and the species, no? Name, if italics are not available, okay? So here we will proceed to the different groups of microorganisms. So these are just basic information or introduction of the types of microorganisms. So we will have more information with regards to the different groups of microorganism during microbial diversity, which is the last topic that we will have during prelims. Okay, so for bacteria class, no, this is considered to be unicellular and belonging to prokaryotes. As you can see here, as I said earlier, we can group bacteria according to the number of flagella or flagellum. As you can see here, we have a bacteria more than one flagella, okay? But what I want to emphasize here, class, is the presence of peptidoglycan in the cell membrane of the bacteria. So if we are talking about peptidoglycan, this is a very important component that can be found in the cell membrane of a bacteria, which enables you to identify whether the bacteria is gram-positive or gram-negative bacteria. Okay? So remember that, class, the peptidoglycan will help you to identify whether the bacteria is gram-positive or gram-negative bacteria. Okay? So I will emphasize that soon. Why? Thus, the presence of peptidoglycan in the cell membrane of the bacteria will give its way for you to classify whether gram-positive or gram-negative bacteria. Aside from that is the means of reproduction of bacteria, which is what we call as the binary fission. So the binary fission is the way of the bacteria to reproduce. Okay, so it looks like a mitosis, class, no? So we have this parent cell, one parent cell, no? Then as time goes by, during reproduction, it will produce two daughter cells. So whatever will be the cellular structures, the organelles present in the mother cell or the parent cell will be the same with the two daughter cells in the end. So that is what we call as the binary fission. Okay, so of course, the motility of the bacteria is by means of their flagella or flagellum. Next is the domain archaea. So what is very interesting with archaea, class, is they are very unique because they can thrive in a particular environment that has a temperature, it's either too high or too low, okay? Then aside from that, they are also classified as unicellular and prokaryotic, no? Aside from that is they can live in absence of oxygen, meaning they can thrive without the presence of oxygen in a particular environment. So they are considered to be anaerobic. Aside from that, if the bacteria has the presence of peptidoglycan, well, the archaea doesn't have peptidoglycan, okay? And then aside from that is we also have different groups of archaebacterial, no, groups. These are methanogen, thermoacidophiles, and halophiles. So all these different groups of archaebacteria are considered to be a unique one because of the habitat that they have. First is the methanogens. So I do not know if have you seen those different news that is available, no, in our local news here in the Philippines, that they got to discover different bodies of water that are capable of producing flame or fire, no? That is because of the presence of methane, no? But despite of such unique habitat, there are microorganisms that can thrive that specific habitat. And this is what we call as Methanococcus, okay? Another is the thermoacidophiles, no? So This type of bacteria can live in extremely hot environment. No, it's not only the very high temperature, but as well as acidic environment. So example is the Thermococcus. Lastly, the halophiles, no, live in extremely high salt concentration, no? So with this, the very high component is the salinity or salt concentration in the water. So there are microorganisms that can thrive such environment like this. And this is what we call as the halobacterium. Next is fungi. As you can see here in our fungi, it looks like a plant, but it is not the same with plants, okay? As I said, fungi is not the same with plants, no? Plants that we can see in terrestrial, no? So as you can see here, we have the hyphae or the hypha. Then we also have the mycelia or the mycelium. The mycelia or the mycelium is the root-like structure of the fungi, no? So basic information or characteristic of fungi is if we have peptidoglycan in our bacteria, in fungi, we can find what we call as chitin, no? Then aside from that is fungi usually described as filamentous, no? It looks like a thread-like structure class, no? And then aside from that is fungi are considered to be important decomposers in an ecosystem. As I said earlier, the bacteria and fungi are very important microorganism in the environment, which plays very important role in decomposing. Aside from that is they're considered to be important not only in terrestrial environment class, no, but also there are also fungi which are present also in other habitats. Okay. Next is protozoa. So as you can see here, the image of the protozoa is different in terms of their structure comparing to the bacteria and fungi, no? So protozoa can be found in different bodies of water, no? Like for example, ponds, no? So protozoa is also called as protists, no? And then this was discovered or first seen by Anton van Leeuwenhoek. Okay, so there are three main types of protozoa, no, according to their structure also. First is the amoeboid protozoa, no. This is the microorganism that is responsible of what we call as amoebiasis, no? So we need to be careful with the drinking water, no? We are using also or drinking. So in terms of their motility class, the amoeboid protozoa were just going to glide, no, in the flow of the water. That is why the amoeboid protozoa is described having false feet because of the structure, but it is not really um literally having its feet, like for example insects or animals that can be found in terrestrial, no, it just looks like having a feet, but it's not. That is why it is called as pseudopods or having false feet. Then we also have ciliates and flagellates because of the presence of this locomotory parts with their body. So for ciliates, these are protozoans having the presence of cilia on their body. As you can see here, it looks like hair-like structures class, no? So this is what we call as cilia. So referring to protozoans having cilia is what we call as ciliates. So this is also the same with protozoa having the presence of flagella, which is what we call as flagellates. But in terms of motility of ciliates class, is the ciliates will just have this vibrating motion because of the presence of the water. Whereas for the flagellates, of course, with the help of their flagella that creates whip-like motion under the water. Then we also have the alga or the algae. They're considered to be aerobic, meaning they need the presence, aerobic and photoautotrophic, they need the presence of light, and energy in order for them to perform their function. As you can see here, if we are talking about algae, class, what comes into our mind is the green pigment because of the chlorophyll, no? And that chlorophyll can be found to what we call as chloroplast, no? And then aside from that is we can find algae in different species, no? Like for example, this one class, this is what we call as kelp, K-E-L-P. So if you're going to see this class, no, for example, via YouTube, is you can see this kelp, very amazing class under marine water, no? So you can see kelp class that is considered to be already a forest in underwater, no? In seawater, I mean. So this is so amazing because the kelp could grow or described as giant kelps. That is why it is so amazing. You can also see other marine life um, which is present in the kelp forest. Right? For example, the sea otters, no? Which is very important guardians in the sea because it helps to mitigate um, bad or threats sea urchins, no, in the coral reef. So the sea otters dwell on that specific habitat, no, the kelp forest in seawater. We can also see other or found other algae cluster. For example, the cyanobacteria, which is the blue-green bacteria considered to be algae. You can also see other algae class, no, whether in terrestrial or in marine, or it could be also in aquatic ecosystem, no? Just like what happened in Boracay class, no? So during the time of the administration of former um, President Rodrigo Duterte, the time that he rehabbed the Boracay, way back, the Boracay is very prone to algal bloom because of the poor sewage that they have. And with that, since they have very poor sewage disposal, is they are very prone to algal bloom. If the water has already an indicator of algal bloom, so therefore it indicates that the water is contaminated with this sewage, no, or perhaps other possible contamination of the water. And when the time that the water is contaminated and the temperature is so high, that is why the algae will bloom. That is why it is called as algal bloom. Okay. Next is the virus, which is in terms of their structure is very unique compared to the other groups, simply because in terms of their in terms of their characteristic, this virus will survive only with the presence of the host, no? So as you can see here, the structure of virus is really different compared to the other major groups of microorganisms I mentioned earlier, no? So what is so easy to identify in the structure of virus is the presence of spikes, no? The red ones, class. So that is why it is called as, for example, the coronavirus, no? Because of the presence of the spikes, no? From the word corona, no? But aside from that, what is so um what is so amazing with the structure of virus aside from the spikes is the presence of enzymes here present in the spikes that helps our virus to adhere, to penetrate to its host, no? Especially if they will be able to identify the whole the immune system of the host is quite weak, no? Yeah. So aside from that is this is the envelope of our virus that helps to protect the genetic material inside the virus, no? So what will happen to our host if ever the virus already penetrated in our system, no? For example, so the virus will going to inject this genetic material inside the body of the host, no? And that's the time how the invasion will happen. So I'm going to discuss about very important information of viruses and how these viruses will invade in our body system during microbial diversity. Okay. Next is we have the parasites. So if we are talking about parasites, we have ectoparasites and endoparasites. So if we are talking about ectoparasites, those parasites that we can see in our domestic animals, no? The lice and the mites, no? So be careful with your cats and dogs class, okay? So aside from that is we also have endoparasites. So these endoparasites could be belonging to cestodes or the tapeworms, trematodes or the flukes, nematodes or the roundworms. So we will have more of this parasites class during finals. So now we will proceed to history of microbiology, no, and the different contributions, important contributions that they made, no, in the discovery of microbiology. So we all know Robert Hooke is very important person in the field of microbiology because he was able to discover what we call as or termed as cell, no? So through observing a slice of cork that he was able to view these little boxes that eventually uh, help him to create this what we call as cell theory, that all living things are composed of cells. Aside from Robert Hooke is Anton van Leeuwenhoek, no? So he is a very important person in the different contributions made under microbiology, no? So according to Anton van Leeuwenhoek, seeing is believing. So it parang to see is to believe sa ating class, no? Na ano, the phrase, no? So si Anton van Leeuwenhoek was able to create, no? Very important lenses and microscope. So with that, he was able to create a very important, no, and powerful microscope in his time, no, that could magnify 300 times. But aside from that, it started when Ewan was able to discover um, different discoveries, no? For example, rainwater coming from clay pot, no? Sa, siguro sa bahay lang nila, no? So he was able to view this rainwater using his microscope. So with that, he was able to view these living organisms, no, from this water using his microscope. Aside from that, he was able also to get samples, no, from men during industrialization era, no, was able to get plaque from these workers in order to see these living organisms. Because during their time class, hearsay are very rampant. So in order for these different contributors, important contributors in the field of microbiology, they need to prove that living things are really existing, no? Of course, with the help of this very important microscope and lenses, they were able to discover that indeed living things are really existing surrounding us. Aside from that, all the discoveries and observation of Anton van Leeuwenhoek was able to send and store in the Royal Society of London. Aside from that is he was also the first person to record faithfully all his discoveries and observations. No, so that is why Leeuwenhoek described bacteria and protozoa as animalcules. No, so these are also the microscopic observation of Anton van Leeuwenhoek, the structures of white blood and red blood cells, no? Also the yeast, the sperm cells, and the bacterial cells. So now we will proceed to different demonstrations class because I said during their time, hearsay are very rampant, no? They keep on debating if this is really existing, no, that time, okay? So that is why there are several important persons that contribute important observation in the field of microbiology. So for example, Francesco Reda, Gianni Tam, and Lazarus Palanzani. So here in the demonstration of Francesco Rede, as you can see here, we have three different jars or container. Then they're all with maybe pork meat, no? So as time goes by, eventually maggots will really present, no, with that particular meat. As you can see here, the first jar is exposed in a very open environment where the flies is also present. Then we also have this another jar which is enclosed with gauze, but we all know that the gauze is not completely sealed, no? But somehow helps also in the formation of or increased population of maggots. The next is sealed jar, no? Okay. According to Francesco Radai, no maggots appeared in the sealed jar. So based on this demonstration, we can say that our surrounding is we may not see these microorganisms class, but as I said, these microorganisms are considered to be ubiquitous, meaning they can be found in anywhere of our surroundings. We may not see them, but there are plenty of them. So that is why that because of microscope, we are able to view them, and indeed there are many of them. That's how diverse these microorganisms are, no? So once your meat is exposed in the atmosphere or in your surrounding, you can expect that it will be contaminated also, no? Another is the demonstration of Needham and Spallanzani. As you can see here, both of the demonstration were subjected into heating, eventually boiling. Both also signified no microbial growth after the heating or boiling process. But in the end, there is already a difference between the two, no? For Needham, after the boiling or heating process, then after some time, the microbial growth recurs again, no? And as you can see also that in the demonstration of Needham, your Erlenmeyer flask is exposed in the open air, no? Exposed, and with that, it can also contaminate your sample, no? So while for Spallanzani, yeah, the same with Needham's, the sample is subjected to boiling or heating process. Then next is same after boiling and heating process, no microbial growth happen eventually until the end. Because for Spallanzani, there is a cork, no, which is placed in the mouth of our Erlenmeyer flask. It could be also cotton plug, no, you can use it, this one. That is why in the field of microbiology, if you're going to isolate microorganisms in the lab, you need to see to it that your samples were not contaminated. That is very important. And aside from that, you need also to practice different techniques, no? While the experiment or the isolation is going on, you need to practice, no? Those different techniques, like for example, aseptic technique, in order for your samples not to get contaminated, no? So that is why in this demonstration, it is very important to secure your sample in order not to get contaminated. We also have Rudolf Virchow, no, in the year 1858, that according to him, living cells arise only from pre-existing living cell, and that is what we call as biogenesis. We also have a demonstration conducted by Louis Pasteur, no, which what he emphasized here in his demonstration is the importance of conducting aseptic technique. Okay, so this is the demonstration of Louis Pasteur. As you can see here, he poured his broth to this type of glassware. According to him, that microorganisms were present in the broth. But of course, class, this is not a direct examination just only because of your bare eyes that you can see microorganisms are present. No, of course. Of course, with the help of microscope, no? So after that, he subjected his broth into boiling. And then as try to notice class the shape of the glassware, where the S-shaped glassware, no, which is at the top of the glassware, where the vapor will come out, no? It is very tiny part of the glassware for the vapor to pass by, no? According to Louis Pasteur, microorganisms were not present anymore, no, because of the boiling process. Next is, eventually, as time goes by, he concluded that microorganisms were not really present anymore after long periods of time. So what we can see here, class, is the importance of aseptic technique, no, that helps getting rid of possible contamination of the sample. Second, the glassware that he used, no, that helps you also to protect your sample from the environment that could contribute contamination to your sample, no? Then lastly, the process also conducted boiling process that helps to protect your sample. That is why in this demonstration, Louis Pasteur emphasized the importance of aseptic technique. So if you are inside the laboratory, you're doing microbiological procedures or experiment, it is really important to practice aseptic before, during, and after the procedure or experiment. Okay. So another important contributions for microbiology is the discovery also of Lewis Pasteur of fermentation and pasteurization. Aside from that, Joseph Lister, which is very important also in the field of nursing, no, referring to antiseptics, no, in treating wounds. We also have Robert Koch, which established the Koch postulates in identifying disease-causing organism. So according to Robert Koch, is he was able to isolate the causative agent of anthrax, which is the Bacillus anthracis, no? So he got this um causative agent from the blood of dead cattle, no? So he extracted the blood and cultured the bacteria, no, inside the laboratory to further examine this causative agent. So what he did, though, in order to prove that there are really disease-causing microorganisms in our surroundings that can give us infections or diseases. So this is what we call as the Koch postulates, that he was able to prove that these causative agents can give us infections or diseases. As you can see here, we have a mice. So what Robert did is, di ba, he was able to extract a blood coming from the dead cattle, no? So what he did is he was able to inject that blood coming from the dead cattle to the mice. And then eventually, this healthy mice, namatay siya, class, no? And then after that is, he extracted again the blood coming from the dead mice, no? In order to culture it. So as you can see here, class, we have this Petri dish with the presence of a media. The media, class, will be our medium in order to isolate the microorganism inside the laboratory, no? So with the help of this media, we can isolate the microorganism inside the laboratory by providing them necessary nutrients in order for them to survive. So that is why we need the media, because this will serve as their substitute environment, because they are not in their natural habitat, no? So that is why we need to provide them a media in order for them to survive. So what Robert did is he was able to pour out the blood coming from the mice to this media. Okay? Then after that, it will not become like this, class, automatically. So after pouring it down here, we're going to incubate that with the help of the incubator. The temperature of the incubator plus the nutrients present in the media will help our microorganisms to grow after 2 to 3 days. Okay? So assuming after incubating it, after 2 to 3 days, Robert was able to prove that there is really microorganism with that blood that he was able to extract it coming from the dead mice, no? So aside from that class is Robert was able to conduct confirmatory tests, no? So he again extracted a blood coming from the dead mice and then to the healthy rat. Then again, the mice is deceased, and then he was able to extract it again, then poured it out to the media, incubate, and then observe. Then again, the same causative agent or the same pathogen that he was able to discover from the first one, no? So therefore, the germ theory disease class is indeed very true that there are infections or diseases that will surface because of causative agents, no, or pathogens. Okay, this is what I'm talking about. That Joseph Lister is very important also in the field of nursing. For example, in the reducing of microorganism in medical setting and preventing wound infections, with help of antiseptics or antisepsis. Then I also included the important contributions of Robert Koch and Louis Pasteur in the field of microbiology. Then we also have here different advanced technology with the help of microorganisms, no? We have biotech, which is very important in the production of food, drugs, and vaccines, no? With the help of microorganisms. We also have genetic engineering that tends to manipulate the genes of these organisms, no? In order to make new products. Or like for example, there are countries that practicing genetic engineering that they tend to manipulate the genes, no? For example, vegetables. We also have bioremediation, no? Because with the use of these microorganisms, they able to help mitigate possible impact, harmful impact, for example, to the environment. So because of the help of microorganisms, it is really possible that it can create positive impact also to the environment through bioremediation. So in summary class, no? So therefore, everyone has microorganisms in and on the body. And this is what we call as normal microbiota or our microflora in our body. Next is the disease-producing properties of a species of microbes and the host resistance are very important factors in determining whether a person will contract a disease. Just like what I said earlier, that, for example, the virus will really tend to be opportunistic pathogen if this virus will able to identify that the immune system of the host is weak. And with that, the virus will able to invade the host easily, no, or contract easily a disease. Then we also have bacterial communities that form slimy layers on surfaces, which what we call as the biofilms. So these biofilms will serve as advantage of this bacteria once they will form as a community, no? Then next is an infectious disease is one in which pathogens invade a susceptible host. So For example, during pandemic time, no, it is very important that we need also to identify those individuals which are considered to be susceptible to these infectious diseases, no? Then lastly, emerging infectious disease, EID, is a new or changing disease showing an increase in incidence in the recent past or a potential to increase in the near future. That is why microbiology is also linked with the study of epidemiology, which is also one of the sciences I mentioned from the start, in order for us also to understand further the world of microbiology. That is why that it is said that after pandemic, there are emerging infectious disease already that are already visible in our communities. That is why there are many researches that we're conducting now, no, proving that there are emerging infectious diseases after pandemic, no. And lastly, class, one of the topics that I am also going to discuss to you during midterms is epidemiology and public health, especially you are nursing students, and you will also have this exposure during your second year in the community nursing or COCAR. Okay, so that would be all, class, and thank you for listening.